Development of functional assays and identification of cellular biomarkers in 3D models is critical because they better represent physiological processes and disease phenotypes. This poster demonstrates our novel breakthrough advancement in biomarker identification in 3D cell models using microfluidic-based automated system. Pu.MA System is a microfluidic-based system that is designed to automate assays for 3D cell-based models like organoids, tumoroids, or spheroids. It is a benchtop instrument that uses a microprocessor-controlled pneumatic system to move fluid between wells via pressure differentials. The system allows automated reagent transfers and incubations, supernatant collections, and in situ lysis and imaging. The key part of the system is Flowchip. Each Pu.MA System Flowchip contains 8 lanes of reagent wells connected by microfluidic channels. 4 Flowchips are placed in holder that locates all wells in a 384 multi-well plate format, providing for 32 samples per assay. Each test lane designated to one organoid or spheroid sample and consists of sample well connected to the reagent wells via microfluidic channels. Flowchip can be filled with any reagents like media, compounds, stains, and etc., depending on the assay configurations. Organoids or spheroids are loaded into the sample well and located in a protected sample chamber at the bottom of the well. This allows reagents to be directed in and out of the sample well without disturbing or drying out the sample. The bottom of the Flowchip is a thin cyclic olefin copolymer film, which makes it compatible with high-resolution imaging. The diameter of the protected sample chamber is 1.2 millimeter, which allows you to accommodate samples of 800 micron to 1 millimeter in size. The advantage of the system is that you can run 3D assays under physiological conditions, avoiding samples damage or loss. All images in this poster were acquired using CellVoyager CQ1 imaging system. It is a spinning disk technology that allows to acquire images using 4 fluorescence channels. The system also supports live imaging under temperature, CO2, and humidity control. The system is integrated with the Cell Pathfinder analysis software, and importantly, it is compatible with Pu.MA Flowchips. On these images, you can see how the Flowchips are positioned inside the instrument and ready for image acquisition. In this study, we used primary cells called TUBCX4IC, derived from a triple-negative breast cancer patient that were created in Dr. Burrow's lab at Tulane University. Primary tumor is characterized as metaplastic subtype, aggressive, and resistant to Taxol therapy. Isolated primary cells were used to create tumoroids by culturing these cells in ultra-low attachment well plate for up to 72 hours. Created tumoroids were then transferred to the Flowchip for further automated drug treatment and assay execution, downstream measurement, and imaging. In our system, we use Matrigel since it is the most used matrix for organoid culture. It is a murine-derived extract containing collagen, laminin, and intactin. We also evaluated an engineered hydrogel called Matrigel, which was developed by the Well Biosciences. It is a xeno-free hydrogel, which is temperature independent, chemically defined, tunable, and reproducible. The solidification of this gel is based on interaction with ionic molecules from cell culture medium. The staining protocol starts with loading your 3D sample to the Flowchip in extracellular matrix. Samples are loaded in a very small volume, forming a mini dome with spheres in it. Extracellular matrix provides structural support and prevents your sample from direct contact with plastic. After loading, all you need to do is place the Flowchips in the Pu.MA system, which will run your assay. Here is the schematics of the complete automated staining workflow. The first step is to load the Flowchips with your samples and staining reagents. Next, you place the Flowchips inside the instrument, select the protocol, and press Run button. Staining steps will be executed by transferring reagents to and from the sample automatically. After the staining protocol is completed, the plate with Flowchips is ready to be transferred to the imaging system for image acquisition. In this study, we investigated the expression of a set of markers. We looked at ecadherin, vimentin, phalloidin, and Hoxt expression. The epithelial-like and low malignant MCF7 cells show intense peripheral ecadherin staining prevalent at cell-to-cell contact areas and show no detectable expression of vimentin. The highly aggressive 4IC tumoroid showed loss of E-cadherin expression while being positive for vimentin. F-actin distribution and organization was detected by phalloidin staining. The staining protocol is valuable for studying the interplay between cadherin cell machinery, vimentin, and associated partners like F-actin, its role in epithelial to mesenchymal transition and metastasis, as well as modulation dynamics by pharmacological agents. We also demonstrated the compatibility of Pu.MA system with other engineered ECMs like Matrigel. Matrigel is transparent and compatible with high-resolution imaging. MCF7 and spheroids show similar E-cadherin expression pattern as in Matrigel. We were able to obtain these high-quality images in Matrigel and Matrigel, demonstrating that Pu.MA system can be used with different kinds of matrices. Here, I'd like to share with you the approach we take to perform cell compartment analysis to look at the expression pattern within each cell. Accurate segmentation can be challenging, especially when it comes to isolation of cell membrane region in 3D models, but provides lots of information about the marker behavior in different cellular compartments. The cell border and cell segmentation here were performed using phalloidin image input. Nuclei were identified using Hoek staining, and cells were partitioned into inner region and outer region, corresponding to the cytoplasmic and membrane regions. Once it's done, we have various graph options to visualize the results. For example, we can quantify membrane to cytoplasm fluorescence intensity ratio. Specific cell populations can be also extracted and classified by getting the feature value data of identified objects. Currently, we're working on optimizing image acquisition and analysis to obtain as accurate segmentation as possible to characterize biomarker expression and cellular localization under various conditions. And to conclude, we demonstrated the capabilities of Pu.MA system to perform complex multi-step protocols like IF staining with 3D cell-based models. The major benefit of the platform are cost, time, resource saving, and minimizing human error while aiding in acquiring high-quality data. In combination with other automated 3D assays, the Pu.MA system enables examining a large set of parameters, including biomarkers, signaling molecules, cell morphological changes, proliferation indices, and toxicity.